Thank you for the introduction. Hello, everyone. I'd like to point out that it was the organizers who chose the title. So I'm trying, since you said I will talk about this and that, so I did my best to put some meat on the skeleton that was imposed upon me. So I looked up HPC, high performance computing, apparently. And what I want to do, so I'll give you an overview of some of the works that we do, and there is a mindset that I'd like you to have. And I'm going to start with showing you a seizure, yeah, and of an epileptic patient. I will not talk only about epilepsy. I will talk about the virtual brain framework and large-scale brain network modeling in general. But then in terms of clinical applications, that's the most developed. So I will start with some words on epilepsy, and I will close the loop at the end again. But this particular seizure, typically, I like to show it because it's, first of all, easy on the eye. Yeah. Second, it is very instructive on multiple levels. So this is what you see a patient here, a patient in Marseille. She's implanted with stereotactic electrodes. Now the seizure starts. Hemisphere implanted, and this part of the seizure, the singing, the rocking. She has a prefrontal temporal seizure. And what you're seeing here, we call this a semiology. There is, it's essentially in behavioral space, a spatio-temporal sequence of, someone talked about primitives earlier, of motor primitives, of behavioral primitives. You see the singing, the rocking, the clapping, behavioral clapping. Yeah? So these are behavioral primitives. The nurse comes in and is trying to interact with her, yeah? Calling her and essentially trying to figure out her degree of consciousness. And the seizure is over now. Yeah? So she's implanted with a set of electrodes. They're about that length. They're so-called stereotactic electrodes, Talarak electrodes. There are either 10 or 15 so-called contacts on it. Let me stop. It stopped, yeah. So contacts spaced 3 to 4 millimeters apart, allowing us to measure the local field potential, the electric field of the neighborhood that is being generated by the neighborhood to sample this. And then typically, a patient has between 7 to 15, up to 20 of these electrodes. So this gives you a sampling of up to 200, 250 points in the human brain. And here you see the time series of the seizure that you have seen right now. Yeah. So these are the behavioral elements. And the time series are plotted here. Clinicians identify seizure onset in these particular areas that correspond to the prefrontal lobe, prefrontal pole in particular, and there you do not have any semiology, any behavioral onset yet. This is coming when the seizure actually starts spreading out and recruiting other areas, and this is something we find very often. There is a distinction being made between symptomatic and asymptomatic seizure, and the asymptomatic very often stays within a small epileptogenic zone and does not propagate. This is not the case here. So the seizure propagates here, coincides with the onset of the behavioral primitives that you have seen, recruits also the part of the temporal lobe, and then reorganizes as time goes by. And the challenge for the clinician is to, on the basis of the SEEG, on the basis of the semiology, which is also a spatiotemporal pattern, as it is being accompanied here by the electrophysiological pattern, spatiotemporal pattern, evolving over time, in conjunction with structural information that we have from MRI, for instance. There are maybe lesions in the MRI, yeah? In terms of, on the scalp EEG, non-invasive, when you have spikes, so-called interaictal spikes between seizures, there are also sometimes not too bad indicators for the organization of this person's epilepsy. So taking all this together, essentially the key question is, what is the epileptogenic zone in which the tissue generates self-sustained, rapid, fast discharges and then propagates to other areas that we refer to as a propagation zone, which may be this or that area, that are actually not pathological. This is not epileptogenic tissue. Yeah? And as opposed to other areas where there is just physiological activity. And there are quite some challenges because physiological activity, I will show you a picture later, in some areas may look as pathological activity if you go into another area. So there are quite a number of challenges that we have to deal with. Spatial-temporal evolution here, spatial-temporal evolution on the behavioral level. I can refer to this as behavior. And if we want to understand this, we have to measure it, yeah? And we have to make the link to the models, which means we have to have models that are capable of generating this type of signatures, yeah, in the human, yeah, on this type of, on this level of organization. And this is one of the challenges. If you talk about integrate and fire simulations, neurons, simulations of, yeah, this does not generate any knowledge on this particular level of organization. We have to go to the system level. Yeah? Links need to be made, of course, to the point neuron or to the, I don't know if you have talked here about the high-dimensional modeling in terms of subcellular arborized dendritic trees. You can go all the way down to this level, as we do it with neuron simulations or arbor simulations, where we have thousands of compartments and where we can use the morphology of the dendrite to generate point neurons that reflect a structure like this. Yeah? But when we want to make the connection to function or behavior, we have to go actually either on the microcircuit, so we have to go to the microcircuit level to link to function, or all the way up to behavior, we have to go to the network level. What is being done is, I think Peter Rolf Seemer gave a talk here yesterday, didn't he? Yeah? So there he was talking about retinotopic mapping, the organization of V1, eventually also V2. So there you have the retinotopic map. Yeah? If you get on this level where you have a parcellation of a brain area, you wash this structure out on the microcircuit level. Yeah? Question, so you lose something, you pay a particular price. And the way how you do this is essentially you start from very detailed models and you map it upon the point neurons. Yeah? So this is being done in the Blue Brain project, where you define transfer functions that are aiding you to characterize this particular network with the appropriate weights. Yeah? That is one reduction. When you go from here to that level of organization, you can either continue doing a fully detailed microscopic bottom-up approach, or you have to apply certain techniques, typically taken from statistical mechanics, mean field theories, or others, to make the step from the networks of point neurons and go up there. And this is when you wash out all this type of information. Yeah? But nevertheless, you're then able to talk about spatio-temporal organizational patterns up here. The question is, do you need this information or not? And for some questions, you may not need it, and this is a direction in which I want to push today. And this is what we've been doing in my lab for the last, well, for a while, years, decades, in fact. Yeah? There are other issues that we have to deal with also. The brain is a distributed system, and if we do these steps across levels of organization, then we have to actually make a commitment to individual regions, yeah? Because there is a detailed region specificity as you go across the cortex. You have different cell densities, you have different expressions on the transcriptomic level. And when you go down to the subcortex or to the cerebellum, you have again a different level of organization. So you have different structures, and on top of this you have a variability that we have to take into account. If you start thinking about a template, then typically this template is the key representation of a brain that does not exist. Yeah? Because you have to map a personalized brain essentially on this template, and like this you change it, you alter it. It's not the same brain anymore. And we know that actually these variations start, they play a role. Yeah? This is ongoing work and will probably occupy us in the HPP for the next 3 years, this variability, and how it maps then on the functional variability. Yeah? And when you go into different areas here, you have an example for a frontal pole, SEG data. And this is physiological activity. This is not pathological activity. So this was recorded from implanted patients that did not have any pathological activity in there. And if you talk to experts, epileptologists that do this type of measurements over and over again, depending on to whom you talk, but I think it's fair to say that they will say we have about 6 to 10 different types of characteristic physiological activity. Yeah? And I picked here 3, amygdala, frontal pole, STG, and what is plotted here is time series. This is frequency over time, and this is the power spectrum. And you see the different types of organizations that you have here. For instance, if you had this type of activity here, if it were in the amygdala or in the hippocamp, it could be mistaken for a seizure activity, whereas here it's absolutely normal. Yeah? So this is the type of information we need to understand. And this mapping that I just spoke about of the variability, of course, it has to express itself in terms of physiological variability. And we never measure this tissue in isolation. It's typically embedded in the network. So there's also a network effect. All this we need to take into account when we go on this level of organization and start talking about propagation of activity through the network. And as I told you, we will wash out this microcircuit aspect and work entirely on the system network level. So for this, I want to give you an overview of some of the neural mass approaches that exist. So in this parcellation in the top right corner that I showed you earlier, a brain region would be typically replaced by a neural mass. Sometimes it's referred to as mean field if it's being derived using some mean field techniques. This does not necessarily have to be the case. The expression neural mass goes back to, what's his name? Neural mass action, help me. He wrote neural mass action, Freeman, Walter Freeman, yeah, in the 80s, yeah? And it represents essentially a population, an ensemble of neurons and their activity that is represented by an appropriate collective variable. The question is already, what is a collective variable? Yeah? I'm showing you some examples here. First of all, this is from Malach's lab, Rafi Malach's lab. This is also human data, intracranial human data, together with an electrode, a microelectrode, where you see the spiking pattern. You can measure also with a macroelectrode the local field potential. Yeah? And then you can actually link it to and compute an envelope function of these two, which is nicely represented here. So you see there are some oscillations going on that are representative of the firing rate and the spike density. So that would be potentially a good carrier of this type of information. Can do the same thing now and in using integrated fire neurons. These are simulations. You can put them in a small network and study them. You put them. So this work that has been done by Damian Battaglia in my lab, you can put. He asked questions: how, to what degree, how can we quantify the information that is being transmitted through the spikes, but still collected by the oscillations using means such as directed transfer functions, Granger causality, etc. This is what you see here: spike raster plots, spikes, time, the density function of the spikes, and then the low frequency spectrum of these oscillations. Key question is, can we replace one of these network nodes here by oscillatory systems and obtain a reduced description? Essentially, this is what I just said here, and this is what we mean by neural masses or mean fields. We have this high dimensional description here that we want to collapse, in this case, into an inhibitory and an excitatory population. And then we need to find a good collective variable that represents the activity of this subpopulation such that we learn something. Yeah? This is an example of the lab of Alain Destec's excitatory neurons, inhibitory neurons, integrate and fire, very detailed, where a stimulus is being introduced, and then the neurons follow. This is simulation. The smooth curve is actually the computed mean field, where he has used a momentum approach, so statistical momentum. First momentum is mean, second momentum is variance, and he can derive equations for that are plotted here in particular, that then reduce, this is the same example I just showed you, reduce the activity to a reduced set of ordinary differential equations, in this case 2 excitatory, inhibitory with nonlinearities, and this is a particular case where he has also a slow variable, so slow ion channel kinetics in there in order to obtain adaptation. Yeah? So this here would be adaptive, integrate and fire neurons in a large population using some mean field techniques to reduce it to a 3-dimensional set of equations per node. Yeah? And here you see the particular reproductions. Some work that actually Lionel in my lab, who's here, and this is his poster, this I took from his poster, he actually took this set of equations and performed a bifurcation analysis of the system using 2 of the dominant parameters, W and nu, that are represented here. And a bifurcation analysis means a system undergoes a qualitative reorganization of its dynamic behavior. This could be, there are very characteristic bifurcations, and there is only a handful, so a finite number of bifurcations, so ways how a system can go from one qualitative behavior, such as equilibrium point, to another qualitative behavior, such as an oscillation. So this you can systematically perform then. So this is saddle bifurcation, this is a Hopf bifurcation, and you can trace out a parameter space, which is extremely useful when you want to start parametrizing this model. The group of Diesmann is then going, Sascha for instance, Sascha von Albada, is then going back into approaches where this type of analysis is being used to parameterize these models from the beginning on, because one of the big questions is, what are the parameter distributions here? Yeah? You can, other approaches that are older, in fact, Alain's work is just a few years old. When you go back in the 70s, for instance, there is basic work put forward by Wilson and Cowan, Hugh Wilson and Jack Cowan, 2 mathematicians that have taken an ad hoc approach, and they said, do I really need to, well, this didn't exist yet, but in principle, do I need to take this approach here? Or can I start with some reasonable assumptions about collective variable is firing rate, yeah? And then I can write down certain equations like the master equation for the experts here that know that. So interactive equations in terms of Lotka, Volterra, type, action-reaction equations, respecting the nature of excitation and inhibition, and arriving at very small dimension equations that then later on by Janssen and Ritt were taken and and put together, for instance, to represent the laminar organization by 1, 2, 3 Wilson-Cowan units, which is nowadays called the Janssen and Ritt model, and very, very often used by people working in brain imaging, typically fMRI, EEG, or MEG. This is one of the standard models that is being used there. Also, in this case, 5-6 dimensional, and again, a bifurcation analysis with phase plane plots can be performed. So this is an ad hoc model, not derived from the bottom up. Now you can take the perspective. Well, here it will become a little difficult to interpret these parameters. You can do that. This is excitatory to excitatory coupling, etc. But they have not been derived. Maybe there is not even a need to derive them, because we know actually, we talked about this variability. There is a huge variability in there, and these details in terms of cellular density, for instance, they may not find any expression in these parameters here. Yeah? Said differently, and here referring, well, to Yves Marder's and Marc Goyard's work, but they didn't say it this way. I'm presenting the work in this way. Said differently, there is a set of parameter distributions, yeah, that give rise to the same behavior of the neurons or of the populations. So these are invariant manifolds, yeah, and they are typically not Gaussian shaped, which is a big problem for all the model inversion techniques that like to infer parameters based on the functional data, yeah, because the majority of those makes a so-called Gaussian parameter assumption, dynamical causal modeling, for instance, yeah, and which you do not have here, obviously. Even worse, please look at this. This banana shaped, if you take the average in this on this co-varying object, this invariant manifold, the center of mass is here. You're off the manifold, which means the system, if you parameterize it under these expressions, gives you a completely off behavior. Yeah? So there some other people then have said, let's say these manifolds do exist. We don't know them. It's a target for sampling. It's a target for investigation, but they do exist. And we can actually formalize them. If they do exist, this type of manifolds, then when we leave from them, either the behavior changes very continuously, yeah, so not qualitatively, or if the manifolds are defined by qualitatively differentiated behavior, then the system has to go in phase transition, has to undergo a bifurcation. And we are looking at the bifurcations on these manifolds, actually. And now the advantage is, once we accept that, we can actually make reference to very rigorous mathematics, bifurcation theory in nonlinear dynamics, and say, well, along these manifolds, these onsets, actually there and only there, the equations are unique and unambiguous. These are so-called canonical equations or generic equations that we can identify. And there we are at the heart of bifurcation theory, and here you have some examples. Where you make a transition or a bifurcation from an equilibrium point to a discharge. This here is the case for 6 of those existing in planar bifurcation theory. These are the most used ones. They can go either from equilibrium to oscillations. Here I write seizure because we are using this for seizure description, or can go back and forth. This one goes only in this direction. This one goes in that direction, so from equilibrium to oscillation, etc., etc. So we can actually take this very seriously and build from this table a taxonomy, for instance, for brain states that start and stop. And here in this case, and in this talk, I'm doing this for seizure dynamics. There is an onset of the seizure and an offset. The onset has certain characteristics. It may start with an increasing amplitude or with a fixed frequency. If I do this, amplitude, increasing, here it's arbitrary, I cannot make a statement, increasing. Here it scales with square root of lambda, which is the distance to the onset. Fixed frequency, perfect, this would fall into this category, supercritical Hopf bifurcation. So we can do this systematically like this. Yeah? There are a few traps hidden behind that, but those I sweep under the carpet for the moment, yeah. Such as you are assuming that you are directly in the generating population and are not having far field effects like a traveling wave, etc. This can be done systematically, and we have actually exploited this for modeling of seizures. And here, this is an in tuto example of the rat. One of my colleagues in my lab, Christophe Bernard, goes, takes a rat, removes the hippocampus in tuto, yeah, and then puts it into epileptogenic conditions. And this here is a seizure in the hippocamp that is being executed. This is roughly a few minutes, yeah, so it's actually a seizure train composed of fast discharges, quiescent phase, pre-ictal state, etc. So multiple minutes. I'm zooming in the second one here, just to show you what this looks like. And here you have the onset, you have zooms in there, so the fast discharges, and what you see very often going towards the end of the seizure is actually a slowing down and the organization in a spike wave complex with the seizure offset to the end. There you can apply then this type of scaling approach as we spoke about and actually extract the information that is in there. But you can also put it in this framework that Alex alluded to, that I will speak about, yeah, that I'm speaking about. Actually here, this is a theoretical, also a model simulation for a particular choice of an entry bifurcation, exit bifurcation. You have here the time series I just showed you. You can look at it from the mathematics of fast, slow systems. For a description of a system like this, you need at least 2 fast variables and 1 slow variable. It's not possible to do differently, yeah? So, and this can be then described systematically. This object with these type of properties, a slow time scale and a fast time scale, is then represented in a 3-dimensional state space, 1, 2, slow, and resembles essentially, this is what we call the fast manifold, fast. Exit bifurcation, this is where the change happens, we're describing by generic equation, and jumps onto the slow manifold, and then jumps again, yeah? These are the key points where the mathematics enters and becomes generic, as opposed to whatever happens here, but there you don't have any qualitative changes. So this can be done systematically, but you need an anchoring point, because as unique as it is, it needs to, like in a Taylor expansion. Taylor expansion is complete, you can compose it, you increase the polynomial order, but you need to anchor it at some point, which you call the operating point, and then you do the expansion. Same thing here. In this case, it's a Tarkins-Bogdanov point. This is parameters. You can describe this by 3 variables and 1, 2, 3 arbitrary parameters that have no meaning at this point. They're dynamic representations that absorb all your physiology that you have in the detailed models. They are in there, but we keep them generic. And then in this parameter space, we can move around and cross through these boundary lines that are the bifurcation lines. And this, the topology of it, this is a homoclinic bifurcation, this is a Hopf bifurcation. The topology of this is generic. This provides us with a navigational map, yeah, in the neighborhood of the Tarkins-Bogdanov point. This can be done systematically, and there is some very neat mathematics behind that that goes actually back to publications of the last 7 years, 2010. There is another one, I'm not showing this here, Beaumoret, but was 2008. So we're talking about mathematics of the last roughly 10 years. Essentially what happens is you map this equation, it has 3 parameters, you span it in 3-dimensional parameter space, you use spherical harmonics, and you essentially show that all the dynamics evolves only on a spherical surface. And you can actually show that the topology on this surface does not change. So you can actually represent everything through 2 parameters. And then you get what you see here, this opening on the sphere you have here. And this here is a generic object, invariant under all operations, said differently. Whatever your favorite physiological map is or model is, it's mappable on this system here, and the Tarkins-Bogdanov point as an anchor is here. And then this is the type of behavior which you can represent by two parameters. So it's extremely useful if you model, but also to sharpen your eye when you go into epileptic data, for instance. Here you have an example in collaboration with Bill Stacy, who has collected actually, by now it's 110 patients. Yeah, that's an older slide. The paper is in revision, submitted, now it's in revision. More than 100 patients, more than 2,000 seizures, and classifying them using this taxonomy that I just spoke about. And this taxonomy had not only scaling properties, it had also properties of bistability, for instance. Bistability implies you can actually stop a seizure when you stimulate it. If it's not bistable, you cannot do this. Yeah? Stimulation is nowadays one of these growing topics in terms of therapeutic interventions. So you better know if you can stop the seizure before you implant a poor patient and have him or her undergo this particular procedure. So this is a societal impact that plays always a role, not a hidden role, a very open role for all of our work. So you can classify the seizures. It turns out that there are two dominant seizure classes. And actually, this type of map allows us to interpret the behaviors that we otherwise would not understand. Here's an example. This is a seizure, again, human seizure, that evolves and seems to stop, and then something happens and it goes out again. Yeah? When you plot its inter-spike interval, you see that it goes, and then it becomes varied, and then something changes, and then in principle it goes up. It actually appears almost as if it changes seizure class. And here what you have is actually, what was it? The amplitude. It goes down, and then it stays more or less constant. This here would be a Hopf bifurcation, and this here would be a saddle node or homoclinic bifurcation. You can actually map this patient's seizure into this map here and trace it out, how the seizure navigates within there, and which actually helps you then to identify where you can interact with that. Yeah? So I went a little bit into detail to show you there is actually some very exciting mathematics behind that, not only limited to seizures, but you can talk about brain states, which is another big topic within the Human Brain Project. Yeah? So ways to deal with that. What we are building here are our nodes, network nodes in these large-scale networks. Can use it either taking a very microscopic bottom-up approach, as it is done by some of our colleagues in the Human Brain Project. There you have to deal with the curse of dimensionality. Yeah? Or you take some of these approaches to go to the neural mass, depending on what you do, like statistical mechanics, ad hoc population, you always end up, here this was the nonlinear dynamics that I was talking about, you always end up with a low dimensional representation thereof. The mean field models typically do not oscillate, yeah? Because you average over firing rates. Those, they do oscillate, yeah? So these are already certain constraints. Having said this, let me show you what one can do with this type of models. Yeah? What I just spoke about, when we are on the large-scale network level, the network nodes become individual neural masses. Yeah? There is another aspect that is actually not known to a large community. The time delays via signal propagation actually play a very important role, especially if the signals oscillate here. And this I'd like to convince you of. First of all, how do we build such a network? For individual patients, we take the DTI, we build the connectome. MRI gives us, reconstructed in three-dimensional space, gives us the topography. This gives us a topology. Together, in the same reference frame, co-registered, gives us a skeleton or an avatar, a biological form that has nodes and links. And in our hands, either we do region-based modeling, then we have about up to 200 of these nodes, or we do what we call surface-based modeling. Then we can, the biggest published right now is about 28,000, where we have to take into account also nearest neighbor, intracortical, within gray matter connectivity, and the large-scale connectome determined by the white matter fibers. Then the mathematics looks like this. Psi dot, depending, so psi is our scalar vector, not scalar, our field variable, yeah, but it can be a vector field, so depending on the dimensionality of the neural mass at location x, this is the neural mass dynamics, intrinsic dynamics. If we do surface-based modeling, we have an interaction integral kernel, typically translationally invariant, nonlinearity, plus white matter connectome, a nonlinearity, and the system undergoes a time delay via signal propagation. And this one is important because in the human brain, the dynamics can, the lengths can be actually quite big. So we have to deal actually, when we talk about the connectivity, the connectome, we have to respect that there is a spatial aspect, the anatomical connectivity, but also temporal aspect, the time delays. Yeah. So, and here these are data from the Human Connectome project. You see that the lengths can go up to 20 centimeters. 1 millimeter per millisecond propagation speed. This corresponds up to 200 milliseconds. Yeah, so this is huge. Yeah, there is variation. There are myelination differences, but we're not talking about 5 milliseconds. Yeah, we're talking about 50, 60, 70 milliseconds. And if the operation frequencies in our brains are, let's say, gamma, yeah, then we talk about 40 hertz. Then we talk about 20 to 25 milliseconds. Yeah, so this is well in this range. Hence, time delays matter. This is a little toy problem that I typically like to show. And rather than dealing with the full connectome, imagine you have a cortical sheet, nearest neighbor connectivity, and one single fiber. This is actually Lionel has a poster on exactly this problem outside, yeah, but doing it with nest simulations. Yeah, I'm taking this here now, doing a mean field simulation. This is the activity, and it evolves over time here. It's plotted. Here you see the simulations, and now I increase the fiber length of this single two-point connection. And the system actually changes its dynamic properties, starts oscillating in space, periodicity, and in time. Yeah. And the only thing that I have changed here is the time delay. And you can actually do this systematically. So this is a space-time plot I just showed you. And if you do, again, a systematic analysis, bifurcation analysis, you can plot the spatial organization and the distance. These are the stationary patterns that emerge, and depending on the distance, which maps upon the time delay via signal propagation, you actually find different patterns and bistability, etc. All the things you expect to find in a nonlinear dynamic system. So, what I just showed you, this particular plot, bifurcation plot, can be performed systematically under variation of different parameters. Let's take this plot here. This is with the mean fields. This is a parameterization, the variability of the thresholds in the neural population, and this is a time delay. And you see the structure here in terms of synchronicity. This is a synchronization dynamics metric for a degree of synchronization that is plotted here. You can reduce this same thing with Fitzhugh-Nagumo neurons, or with a full network of edX integrate and fire neurons in NEST. Yeah? So this is at the end of the day exactly what we want to have. Yeah? On this type of level of organization, we'd like to map out the parameter spaces for the full network. And this we can do for the full network by essentially rescaling the connectome. Very quickly, let me show you that you can actually quantify the connectome on this level of organization by taking the time delay into account and putting a so-called phase oscillator, a chromotor system, on each of these network nodes. And this just tells you this is a rotating phase like a discharge. Yeah? When you do this, and the time delay coming from the connectome enters in there, and with some very basic pedestrian mathematics, you can actually demonstrate that this time delay goes into the coupling, omega ij, which is a space. I call this a time, and this I refer to as a space-time structure of the connectome. You can reduce it by absorbing the time delay in here, and now you have both information, the time delay and this in there, the connection. And here in the toy network, with no time delay, this would be the connectivity, where the thickness indicates the strength. And in the rescaled connectome, as a function of the distance of the time delay, you can see that it can completely change. And this can actually change even in full connectomes here coming from the HCP, which are typically characterized using graph theoretical measures, yeah, such as in strength or the dynamic core. But when we do this, they become frequency dependent, and these subnetworks that determine the overall behavior within the system become frequency dependent objects. And the strengths to which individual areas respond become frequency dependent in terms of the interactions through the networks. This is a form of thinking that currently does not exist in the community because the time delays have not been considered systematically. This is a very quick other example. Here you can see the phase clustering within a connectome-based network of all the regions oscillating at 10 Hertz, 20, 40, 50, and 80 Hertz. Here you have the functional connectivity and the phase clustering index across all the regions within the connectome. And you see, as a function of the frequency, how the system can completely reorganize itself, even becoming completely flat in the lower beta range. Yeah? So, space-time structure of large-scale networks matters. It does not matter typically in microcircuits. Here we have to take it very seriously because it can be at the base of diseases. It can be at the base of our integrative capacities in cognition when we want to, for instance, perform a multisensory integration. It is exactly where we use this type of mechanisms. Everything I'm going to tell you, and I have told you, can actually be reproduced in, I'm changing gears now. I'm going to applications. System, architecture of the virtual brain. This is a software written in Python. Yeah, there's going to be another slide. The virtual brain is one of the tools in the Human Brain Project. It has happened, it precedes the Human Brain Project. Yeah. We have made the first release in 2012, but now it's going to be integrated into the joint integrated platform of the Human Brain Project, because the tools that we need here is we need to link them to human brain imaging signals. Yeah? So we need the organization and we need the 3D reference frame in order to embed our networks into these structures and compute the forward solutions. So currently it's available here or directly in the collaboratory. It runs on laptops, but you can also put it on a high-performance cluster, which we do together with our colleagues in Jülich, for instance. At its core is a simulator that we have optimized to run with time delays. Yeah? And that was one of the issues. First, the simulation engine is agnostic towards the population or neural mass model. It can either evoke stochastic or deterministic algorithms for numerical integration, and it can generate the type of brain imaging signals using state-of-the-art forward projections for EEG, MEG, fMRI, etc. SEEG also, which I will show you. And then you can implement different types of pipeline, or their pipelines are there to read in from MRI data, structural connectivity, cortical surface, or other types of data. Just a few snapshots for EEG, MEG, and fMRI. Here we use actually 3 models for the Bolt monitor. This is the balloon Windkessel model and the SEEG, because each of these electrode contacts samples from a particular volume around it. This needs to be taken into account. Some snapshots of the interface. There is a graphical interface, but most of us use it in Python notebooks, yeah, directly from the command line, so you can manipulate the connectivity. And the surface, the different type of renderings you can perform and compute the type of time series, either directly on the source space or on the EEG, here scalp EEG for a particular subject. This can be done also for the mouse, which you find in there, which has advantages for the mouse brain. You can put it in the scanner, but you can use also the Allen Institute database. 5 minutes, thank you. Yeah, and there are some applications. I'm just going over this. This has been applied to perform some work in healthy aging. Yeah, basically based on your fMRI signatures, you can extract your age, or to issues such as stroke in Alzheimer. And what you see here is essentially what people have done. This gets me to the last few minutes of my talk, they have chosen a particular metrics. They take the model, they choose a metrics, a type of behavior of the researcher believes it's important, such as functional connectivity, yeah, which is essentially the set of covariations between the areas as averaged over time. The question is if that metric is informative or not, yeah? This is not a clear, there is no clear answer to this. At least you can publish with that, yeah? And then you can plot parameters and use this metric and extract parameters for the model and see if these parameters for the model are predictive for patient stratification, for instance, yeah? It is for stroke, it is for Alzheimer. Example, you have a stroke. You go through a scan after the stroke, you fit the parameters, and before therapy, the fitted parameters can predict, at least statistically, if your rehabilitation will be successful or not. And this is actually very important because it's quite some effort for the patient to go through a rehab. Yeah? So one may consider other approaches. So it works for this, but there are many questions that are open. The best work example for the moment is epilepsy, and where we do the following. In epilepsy, we have a network data feature, which is actually the propagation. And this is how I started, if you remember. The activity propagating through the network. It's a spatiotemporal pattern. This is, of course, supported by network approach, as I presented to you. On top of this, there is a good data fitting target. It's the epileptogenic zone that is represented in the parameters of the network. Yeah? We can fit that. Yeah? And then it is linked to a network intervention. Yeah? Once we have the epileptogenic zone, it can be operated, it can be surgically removed. So we can actually do the inter-pipeline, which we call the virtual epileptic patient, and go from imaging of the patient, recreating his, her virtual brain, equipping it with the epileptic neural mass model, fingerprints, so data fit the patient, and make predictions about what epileptogenic zone to resect for this particular patient, so to go through surgery. And this, in the remaining few minutes, I'd like to show you how we do this. This is a patient from Marseille. We have reconstructed her brain. And in addition, what we sometimes, we can refine the parameters. This woman had a hypothalamic hematoma, which in the hypothalamus, which is a cell cluster essentially, and the question is where it is located, but it's a lesion that is visible in the MRI that biases our parameters. So we're not completely blind, yeah, when we do this. And then we can set up her virtual brain models. These are all the sources, in this case about 200. And the advantage of this is because we start with structural data, we are every other approach starts with functional data, but we start with structural data, which means we can put in electrodes where we want, amongst others where the epileptologist placed the electrodes. But a big issue in epileptology is also the missing electrode problem. What if I had placed another electrode in here in this area? Would I have made, as a surgeon, a different surgical decision? Yeah? So this problem we can address, and we are working with our surgeons on this. And here you see the patient I just showed you. After her seizure starts on the right hemisphere. This is simulated data though now. These are simulations. Starts on the right hemisphere, propagates through the thalamus, callosal fibers on the left hemisphere, recruiting temporal lobe, left parahippocampus and hippocampus. Yeah? This is her seizure recruitment pattern. And these are her data, right? Sometimes the seizure stayed on the right hemisphere. So this is, I'm zooming into the particular seizure. And this is the seizure pattern I just showed you in the simulation, starting on the right hemisphere and recruiting the areas in the same electrodes as we just had it. The data feature we use, this I need to skip. I need to do very quickly for running out of time. It's essentially, these are the data. We compute the envelopes, and it's a seizure propagation pattern of the envelope that we fit using Hamiltonian Monte Carlo techniques. And please note, all I want to say here at this point is Monte Carlo is standard in astrophysics, in elementary particle physics. It's beyond state of the art at the moment in neuroscience, and we are trying to adapt it to this type of problems in neuroscience. I don't have time to say more about this. All I can tell you is that we are fitting the epileptogenic zone using Monte Carlo techniques, and here you have the parameter distribution of the epileptogenicity parameter per network, and this is the information we are giving back to the clinicians. And when we do this, giving it back to the clinicians, then we compare it with the epileptic surgery that has actually been executed. This is what we see here. In white is the epileptogenic zone, in red is the propagation zone, and in green is the difference area between virtual brain and what has been actually executed in terms of the surgery. When we map this size of the green areas, whether it's additional or missing area, against what we call the angle score, which is surgery outcome, we find actually that the failure of surgery, according to angle score, is associated with a larger area of this green, yeah, or with a discordance of what we what is coming out of the virtual brain score, yeah, yeah. So this holds statistically, and we are designing a device at the moment, a whole clinical score, to perform this actually in a clinical trial with 400 prospective patients. And essentially this is happening together with a company, also Dassault System, that some of you may know from the CAT software. Yeah? So they are building essentially the pipeline that I just presented you, and we are using it in the clinical trial where half of the patients will go through the virtual epileptic patient software and the others not, in order to being performed. And by the end of 2022, we should have the first results coming out of this. Yeah? So this is our way of clinical translation. And let me stop here. Thank you very much for your attention. Yeah? That was a large tour I wanted to give you. Thank you.